Okay, we're going to talk about organic macromolecules for this lecture. So, organic macromolecules, when you think of organic macromolecules, it is carbon containing molecules, right? So, carbohydrates, lipids, proteins, nucleic acids. Enzymes are sort of they're a category of proteins, and then ATP is a modification of nucleic acids. I put in micronutrients too because I wanted to talk about a little bit about metabolism and the requirements for processing, your carbs, lipids, proteins, um, and you need micronutrients to do that. Okay, so let's do the laser pointer. Okay, so what are organic macromolecules? Organic compounds are comprised of carbon, hydrogen, oxygen, and sometimes nitrogen if we're talking about proteins. They're found in living organisms and they come in four categories. Carbs is one, or carbohydrates, sugars in humans, and starches in plants. Whoops. I'm trying to find my stylus and I grabbed a pen instead. Okay, then there's lipids. So lipids, the category of lipids includes your fats, your phospholipids, and your steroids, right? Proteins, proteins are made up of amino acids, and then nucleic acids comprise RNA and DNA. These molecules are called macromolecules. Why are they called macromolecules? Because they're composed of subunits called monomers. Monomers join together to form polymers. So when we talk about the monomers, we're talking about the lowest structure, and then these guys are combined together to form carbs, lipids, proteins, and nucleic acids, or polymers. All right, so let's look at some basic reactions. You covered these in chemistry and you might not have realized that you were going to apply them to here. So let's talk about this one. And I don't want you to know all the complex steps. I just want you to know that a dehydration reaction is used when you're synthesizing a polymer or when you're trying to make a longer chain, right? So in this class, we primarily focus on theory of things. I want you to understand how things work and why we do things. Okay, so here's a polymer. Let's say this is 3 glucose molecules, right? And then we want to add another glucose or a fourth glucose onto that. How do we do that? In order to do that, we need to remove water. So we're going to remove the OH here and the H here. So we're pulling off water, hence the name dehydration, right? And we're forcing a bond there to grow the polymer. So this is now 4 sugars long instead of 3. That's a dehydration synthesis reaction, or condensation, right? So there's a lot of different names for one reaction, of course, because one name would just be too easy. Hydrolysis. So when do we use hydrolysis? Hydrolysis, first off, means adding water, right? And when you add water, you're actually breaking a bond. So here's 4 glucoses, right? Polymer 4 glucoses. And let's say we want to snap this glucose off. You're going to add water, and don't forget that there's an enzyme here. So the arrow, even though we're not seeing enzymes, there's enzymes here, and we'll talk about enzymes in a little bit. So you're adding water, and so that's called hydrolysis. Hydrolysis, if we look at the name hydrolysis, water and lysis means to split, right? So you're going to add water and you're going to break that bond. And so you're taking that 4th carbon off, or excuse me, 4th sugar off. Okay, so carbohydrates. So the first category that I want to talk about are carbohydrates. Those are complex carbs and sugars. The main carbohydrate for our body is glucose. We can use fructose, and we and our cells will actually convert that to glucose. It's a major source of energy for cell metabolism. Your body uses carbohydrates first for fuel, so I want you to know that. The chemical formula of all carbohydrates are 1 carbon to 2 hydrogens to 1 oxygen. So if we're talking about glucose is C6H12O6, right? So it's a 1 to 2 to 1 ratio. Where do we get the energy from? So the energy is actually stored in those carbon to carbon bonds. So as we snap off or as we break those carbon to carbon bonds, you're going to grab that energy and stick it on to make ATP. And we'll talk about that once we get to metabolism. Glucose is a monosaccharide. What does monosaccharide mean? Mono means one, saccharide means sugar. So it is a single ring structure. I don't expect you to know how to draw this, right? But just so you have an idea of what glucose looks like, this is the most common form in the body. This is a three-dimensional, and this is a stick figure, or the line model. I forget what the technical term of it is. So this is your first carbon, your second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. And it is a six-ring or six-sided ring, so it's a hexose, right? So this is glucose, and it is a hexose, and this is considered to be a monomer or a monosaccharide. Simple carbohydrates. So monosaccharides are simple sugars. Disaccharides, which is on the next PowerPoint slide, are simple sugars, and then everything else is a polysaccharide or complex carbs. So monosaccharide, simple sugars with 3 to 7 carbon atoms. I want you to know these three. So glucose is a 6-carbon hexose. This is what glucose looks like, right? And galactose is a 6-carbon hexose as well, and the structure is a little bit different. Where is it different? It's different at the 4th carbon. Okay, you don't have to know it for an exam, but if you've taken organic or biochemistry already, it's right there. Fructose is found in fruit, and it's found in the male reproductive tract for the sperm. It is a 6-carbon pentose. So here's fructose. Here's your first carbon, second, third, fourth, fifth, and sixth. And it is a 5-ring structure, so we call it a pentose. Okay, so these guys are examples of monosaccharides. Disaccharides, what are di? So, or what does di mean? Di means 2 saccharide sugars. So it's 2 simple sugars stuck together via dehydration synthesis reaction. I want you to know that sucrose is glucose and fructose stuck together. So this is an example of sucrose. So table sugar is an example of sucrose. Is this easy? Here's a picture of it. Fructose bound to glucose via dehydration synthesis right there. Is this easy to break down or hard to break down? Right, it's actually a simple sugar, so you just have to break this one bond, so it's really easy to break down. So simple sugars are good when you're climbing up a mountain and you need quick energy because they're going to be easy to digest. If in your daily life, simple sugars are not so great to have, you want to limit those because they're so easy to digest. They're going to spike blood sugar really quickly, right? You actually start breaking down sucrose in your mouth. So if you've ever stuck a cracker or a piece of white bread or a sugar cube or what have you in your mouth, you're already starting to degrade it. So, evolutionarily, we were designed to break down sugars or use sugars quickly. And evidence for that is you taste sugar on the front sides of your tongue and you start breaking it down in your mouth. Okay, so let's see. Maltose. Maltose is glucose and glucose stuck together. And then lactose is glucose and galactose stuck together. Where have you heard lactose before? Good, it's in dairy products. So it is actually a milk sugar. So you find lactose in mammalian milk, right? If somebody has an issue with breaking down lactose, they're called lactose intolerant, right? So lactose is a milk sugar, and lactase is an enzyme that actually breaks down the lactose in your small intestine. And if you don't have enough of that enzyme, then you can't break down the lactose. And then the bacteria in your small intestine go crazy breaking it down, and they produce a lot of gas and cramping and diarrhea, and it's not fun. This is different. So lactose, this is different than an immune response, having an allergic reaction to this, right? An immune response means your gut is having an immune response to lactose. All right, so how do we use these guys? So here's sucrose, right? We're going to use sucrase enzyme or amylase enzyme. ASE denotes that it's an enzyme. add water, so hydrolysis, right? Add water, and you're going to rip apart that bond. So to make glucose and fructose. So where does this happen? This happens in your mouth and in your small intestine. Then these two guys, your glucose and fructose, are going to get pulled in from your small intestine into your bloodstream, go to your hepatic portal, right? To your liver, and then get dumped into ultimately the inferior vena cava to circulate around. Fructose is going to get converted to glucose, and so this is the your main blood sugar in your body. Complex carbs are called polysaccharides. These are many monosaccharides stuck together, condensed by dehydration synthesis, right, to form a polymer. So many of these monosaccharides stuck together are called a polymer or a polysaccharide. These are good to have in your diet because they take more time to digest. They're not going to spike your blood sugar as quickly, and they're going to give you sustained energy throughout the day. So can you think of a polysaccharide or a complex carb, right? So wheat bread is a typical one. Brown rice is another typical one. When you think of, legumes, beans are polysaccharides, anything that's going to have a complex carb. Veggies, right? And some veggies, well, most veggies. Okay, good. So you want to have more of them in your diet, or more of those in your diet, excuse me, because they're going to take more time to digest. I'm really tired today, you guys. Glycogen is a storage form of glucose in animals. It's stored in the liver and skeletal muscle, right? So glycogen is actually a chain consisting solely of glucose, and it's stored in the liver and skeletal muscle. And we'll talk about that in a second. Starch is a storage form of glucose in plants. It's stored in the roots, right? And so we eat the roots and we get the starch from the plants. Cellulose is part of the cell of the plant cell wall that we can't digest. And so cellulose is a complex carbohydrate, but we don't have the enzyme to digest it or break it down. So it ends up being roughage, and it helps our digestive tract to keep moving. Right, here's a picture of glycogen. So notice that it's a glucose molecule bound to a glucose, bound to glucose, bound to glucose, bound to glucose. So it's highly branching. It's a huge molecule of glucoses just bound together. And so this is glycogen. It kind of looks like coral, if you will, right? So this is stored in your skeletal muscle and in your liver. Your skeletal muscle is going to store this and use it to make ATP for skeletal muscle contraction. So it doesn't share it very well. You can pull it from the muscle, but it's not as effective as the liver. The liver stores this to regulate blood sugar. So when your blood sugar is low, let's say when you're sleeping at night, or if you haven't eaten, in 8 hours, your liver is going to snap off. It's going to break this bond, and it's going to dump this glucose into the bloodstream so that your body can use it. It's going to snap this one off, dump it in the bloodstream, snap this one off, dump it in the bloodstream. And of course, this happens with a lot of them, right? So if blood sugar is low, your liver is going to break off glucose monomers from the glycogen molecule to regulate blood sugar, to bring it back up. Okay, here's a picture of what starch looks like in potatoes. And so notice it's sort of this helical form. And then this is what glycogen looks like in skeletal muscle. And I like how this is a chicken wing. It's a fried chicken wing. But if you think of skeletal muscle in animals, skeletal muscle has glycogen, and so does the liver. Cellulose is insoluble dietary fiber, keeps your digestive tract moving. So we just talked about that. Soluble dietary fiber is important to have. You can find it in oat bran, legumes, fruits and veggies, right? Why are they important? Because they slow down digestion. When these food sources hit the small intestine with water, they turn into sort of a gel, and they decrease the absorption of cholesterol and fats into the bloodstream. And some of them actually break off and go into the blood vessels and help protect those blood vessels, okay? Here's a picture of cellulose. Again, this is not we can't break this down. Soluble dietary fiber we can break down. And it's good to have in your digestive tract. So comparison table. Simple carbs, they're digested quickly. They're mono or disaccharides. Complex carbs are digested slowly. They take way longer to digest. They're polysaccharides. Simple carbs do not contain additional nutrients. That's not quite true. So if we're talking about fruit, it does have vitamins and minerals. But if we're talking about table sugar or any like white breads or what have you, you know, they're kind of like wasted calories, right? If you eat a donut or a piece of white bread, you're not getting in very many nutrients compared to eating complex carbs, right? Complex carbs have a lot more, they're more nutrient dense and they're going to have a lot more vitamins and minerals. So examples of simple carbs, table sugar, fruit juice, milk, and yogurt. Again, if you have the choice, you're going to pick fruit compared to fruit juice because you want the fiber and all the other nutrients, not just the juice, the sugar portion of it. And then complex carbs, breads, whole grain or whole wheat breads, you know, beans, nuts, pastas, especially the whole wheat pasta or the bulgur, and the vegetables. Okay, the next category are lipids. They're made mostly of carbon and hydrogen atoms. They're partially or entirely hydrophobic. What the heck does hydrophobic mean? Okay, so I want you to take note because this can get confusing. I know you talked about this in chemistry. So hydro means what? Water. Phobic means fearing. So fats are water fearing. Fats are also lipid loving or lipophilic, right? So let me see if I can write that down. Okay, so hydrophobic, that means what again? Good, water fearing. They are also a lipo, L-I-P-O, philic, P-H-I-L-I-C. What does lipophilic mean? Fat loving, okay? So what does this mean? They're hydrophobic, which means they don't like water, so they're not gonna dissolve in water. They're lipophilic, so because they love lipids, they are going to dissolve in lipids. So for example, if I have something that's a lipid, it would dissolve in olive oil, right? If I put like butter or olive oil with water, it wouldn't mix. And so you learned in chemistry, let me write it over here. Like dissolves like dissolves. So something that is polar like water is, water is going to dissolve something that's polar or charged, right? So polar would be like glucose, or ions are charged, right? So sodium or chloride. Something that it is fat soluble is going to dissolve fats. Okay, so keep that in mind. Let's see. So some have polar groups on one end for fats and non-polar groups on the other end. Polar means what? Charged or slightly charged? Okay, so slightly charged, right? So one side is slightly positive, one side is slightly negative. They don't have a true charge, like water. So if you look down in the right-hand corner, these little guys are water. They're slightly negative on the oxygen side and slightly positive on the hydrogen side. If you don't remember what polar is, you need to look it up on the internet. And non-polar, look that up as well, right? So some have polar groups on one end and non-polar groups on the other end. If that's the case, we call them amphipathic. Detergents, so laundry detergents work this way, and soaps, hand soap works this way. They have a polar group which dissolves in water and a non-polar group which dissolves in the dirt or oil or the stain in your shirt, right? And so the nonpolar attaches to the oil stain in your shirt, and then the polar dissolves in water and it takes the stain away. You're going to use lipids as a fuel source after you exhaust carbs. Okay, so use carbs first as a fuel source, lipids second, and then proteins last. Carbs provide twice the energy, lipids provide twice the energy that carbs do, so we need less. I'm going to talk about the nutrition assignment in lab. So if I haven't talked about it yet, you know that carbs give you 4 calories per 1 gram of carb, and fats give you 9 calories per 1 gram of fats. And so fats give you twice the energy, so you don't need to have a lot of them in your diet. Here's an example of a fatty acid, and here's an example of an amphipathic or a polar and nonpolar fat. So here's the polar head, and here are the nonpolar tails. If you take a bunch of lipids that are have polar heads and nonpolar tails and throw them into water, they will orientate themselves so the polar heads are facing the water and the nonpolar tails are isolating themselves. So this ends up being a nonpolar region or a lipid soluble region, and these guys are the polar region interacting with the water. Lipids. So lipids are not water soluble. So when you're trying to get these guys transported in the bloodstream, when you are trying to get them transported in the blood, blood is made up of mostly water, and so these guys aren't going to dissolve in the bloodstream. So you have to have some type of lipoprotein transport them. We have low density lipoproteins, we have high density lipoproteins, we have very low density lipoproteins. And then we have the free fatty acids, which is a breakdown of some of the lipids, and these guys are transported by albumin, and albumin's made in the liver. Examples of categories of lipids: triglycerides, phospholipids, eicosanoids, and steroids. So these are the categories of lipids, and we're going to talk about each of them. Triglycerides. Triglyceride. Tri means 3, glyceride, so it's a glycerol backbone plus 3 fatty acid tails. It's nonpolar, so they're transported in the bloodstream with using very low density lipoproteins, so VLDL. These guys are what we typically think of as fats, right? So when you cut into a steak and you see the marbling or the fat, that's the triglyceride. It's what we actually store in our fat cells or in our vacuoles in our fat cells as energy reserves. We break down those fatty acid tails and the glycerol backbone for fuel. These guys have 4 important functions. They're used for fuel, obviously. They protect internal organs. They provide insulation, so we lose less heat to our environment. Another really important one that I want you guys to know, especially for the exam, is attract and store lipid-soluble vitamins. I want you to memorize lipid-soluble vitamins. So lipid-soluble vitamins, the only ones that we have are A, D, E, and K. All right, so vitamins A, D, E, and K, since they're lipid soluble, they need lipoproteins to be transported in the plasma, and they will be stored in your body fat. So, because they're lipid soluble. All the other vitamins are water soluble, so they're just going to dissolve in the plasma, and then you're going to excrete them out in your urine or pee them out, right? So let's say I take a bunch of vitamin C. Do I have to worry about overdosing on vitamin C? No, I don't, because it's water soluble, and so I'm going to excrete it out at the kidneys, right? Let's say I'm deficient in vitamin D, which most of us are in the United States, and I decide to take excess vitamin D. Do I have to worry about overdosing? Yes, I do, because vitamin D is stored in my body fat. It's not going to be excreted very easily. And so you have to be aware of overdosing on these guys. There are certain drugs that are stored in the fat, right? And then there are certain toxins like mercury and DDT. Mercury is so dangerous because it's stored in your body fat, and it is stored in your reproductive organs, as well as DDT. And so you can accumulate these toxins very easily. DDT is a pesticide. It was used in the 1950s. It affects reproduction. It really affects reproduction of birds. So it makes their shells soft. So their egg shells soft. So when they sit on their nest, the eggshells break. And so a lot of birds, the populations dropped significantly in the 1950s. And then DDT was banned. And President Trump has authorized DDT as a pesticide. I think it was last year. Again, it's really dangerous because it has serious reproductive issues in our bodies, and it stays in the soil for a really long time. So if you look at soil, you can still find DDT today in that soil. So it doesn't break down very easily. Here's the triglyceride structure. So here's the glycerol backbone plus your fatty acid tails. Okay, these fatty acid tails are just change of hydrocarbons. What the heck is a hydrocarbon? Carbon bound to another carbon, bound to another carbon, bound to another carbon with H's off of it. Here's a picture. So here, the bottom one is a triglyceride, and then the top one is breaking it apart, or this is forming it, whichever way you want to look at it. Let's look at the bottom one. So this is a triglyceride. Here's your glycerol backbone. And then here is your fatty acid tail, right? So the COO is not the hydrocarbon. It's the carbon bound to the carbon, bound to the carbon, bound to the carbon, et cetera, with the H's off the sides of it. So this is a fatty acid tail. This is another fatty acid tail. And then this is another fatty acid tail. And to use these guys, we'll transport them in the blood using very low density lipoproteins. It actually goes from the digestive tract to the liver, and then the liver processes these guys somewhat, and then the liver dumps it into the bloodstream using low density lipoproteins to get to the tissues where you store them, or to the fat, or to whatever tissue you need. Take a look at the top one. So notice the top picture says this fatty acid tail is saturated, this second fatty acid tail is saturated, and this third fatty acid tail is unsaturated. What the heck does that mean? If you go back to your chemistry, the carbon bound to the carbon bound to the carbon, notice those are all single bonds. That means each carbon is saturated with the maximum number of bonds, right? So this has 4 bonds, that has 4, that has 4, that has 4, that has 4. So each carbon has a maximum number of bonds. The fatty acid 2 in this picture is saturated. Fatty acid 3, take a look at the 1, 2, 3, 4, the 4th carbon. Do you see how there's a double bond there? This carbon, the 4th carbon, has it's bound to 3 things, not 4. And then this one is bound to 3 things as well, not 4. And so because it's not bound to the maximum amount of hydrogens or carbons, it's called an unsaturated fatty acid tail. Another short way to explain this would be if you see a double bond, that means that it is unsaturated. Saturated means saturated with hydrogens. Unsaturated means that there's a double bond there. And I'm sure you learned that in chemistry. You might not remember it. But when you have a double bond, it's unsaturated, okay? How many double bonds do you see in this carbon to carbon tail? I just see 1. So this is mono unsaturated, okay? Does that make sense? Mono unsaturated. What if we had many double bonds? It would be polyunsaturated, okay? So we'll get to that in a second. Fatty acid tails, they're long chains of carbon and hydrogen with a carboxylic acid group at the end. So carboxylic acid can be a COOH or a COO minus. Either one is considered to be carboxylic acid at one end, right? The hydrocarbon chain is nonpolar. This carboxylic acid group is polar. So here's a fatty acid tail. This is lauric acid. Here's your hydrocarbon chain, right? Carbons with hydrogens off of it. And then here's your carboxylic acid group, or your carboxy group. This is a C double bonded to an O to an OH. And again, this could be COO minus, and it would still be the carboxy group. This is actually lauric acid, and this is what it looks like. Fatty acids, breaking that carbon to carbon bond yields energy. And since we have more carbon to carbon bonds than in carbs, we're going to make more energy, or we're going to get more energy from that fat. Most of the fatty acids we can synthesize, but some we have to get from our diet. If we have to get them from our diet from an external source, they are called essential. Right? So essential fatty acids are fatty acids that we have to get from our diet. So these guys are called essential, ESSENTIAL, right? So you've heard the term essential fatty acids. Do you guys remember which ones are essential fatty acids? Okay, we'll get to that in a minute. How do we characterize these guys? We can characterize them. We can say they're short, medium, or long chain fatty acids, and by the number of double bonds. So saturated, how many double bonds does it have, guys? None. Good. Unsaturated. So monounsaturated means one double bond. Okay. Polyunsaturated means many double bonds. Okay. Essential fatty acids. Here we go. So essential, what does essential mean? We have to get it from an outside source, from our diet. We can't synthesize it, right? So essential fatty acids, it's important that you have these in your diet because you can't synthesize them, all right? Omega-3 fatty acids, which you've heard, omega-6 fatty acids are essential, meaning they must be obtained from the diet. Omega-3 fatty acids are associated with healthy brain and nerve functioning and decreased body inflammation. Omega-3 fatty acids. Here are your omega-3 fatty acids. So EPA, which is eicosapentaenoic acid, DHA, which is docosahexaenoic acid, and ALA, which is alpha-linoleic acid. These are your omega-3 fatty acids. You need them because they help lay down the fats, the insulation of the myelin sheath around your nerves in your brain. And you want a high enough level of omega-3 fatty acids because it's going to decrease general body inflammation, right? Where do we find these guys? You can find them in cold water fish like salmon and sardines. A good place would be flax seeds, walnuts, and avocados, okay? So where have you seen DHA before in relation to babies? I have a 7-month-old right now. So I'm all about making sure he gets enough omega-3 fatty acids. So DHA is found in formula. It's also found in prenatal vitamins because you want to make sure you have enough DHA for the fetus's brain development. And right now, we're giving him avocado. So since he is 7 months, he can have pureed stuff. So we're actually pureeing avocado for him. Avocado has a lot of alpha-linoleic acid. It doesn't have usually docosahexaenoic acid. DHA, you have to, not all the time, but you have to get it primarily in cold water fish. A lot of the vegan sources, like when you think of flaxseed, walnuts, nuts, seeds, and avocado, they have ALA. So you just have to be aware of, if you're vegan, what types of foods have omega-3 fatty acids and which one. Okay, omega-6 fatty acids are found in primarily animal meat. So excess of these guys are associated with inflammation. We get enough omega-6 from our Western diet. We need to get more omega-3 in our diet for Western civilization, for Americans. Okay, so saturated versus unsaturated. We talked about this, but here's the PowerPoint slide. So your fatty acids or fatty acid tails can be saturated. That means there's no double bonds between the carbons. All carbons have the maximum amount of hydrogens bound to them, or they can be unsaturated. Unsaturated means that there's one or more double bonds in that fatty acid chain. Monounsaturated, one double bond. Polyunsaturated, many double bonds. Here's a zoom in of that. So saturated, these carbons are saturated with the maximum amount of bonds. So this carbon, the maximum amount of bonds it can make are 4, so each one has 4 bonds, right? Or it's saturated with hydrogens. Unsaturated means that you've snapped off 2 hydrogens and you forced a double bond there. So unsaturated is a carbon double bonded to a carbon. It means it's unsaturated with hydrogens, or it has less hydrogens than it could potentially have. Why is this important? So if you look down, it forms a kink in the chain. Okay, so why is this important? This double bond right here is going to make this fat kink. And so it forms a kink in the chain of this fatty acid tail. And when it kinks or bends, this is going to increase the fluidity. So it's going to increase fluidity. And so the more double bonds you have, the more fluidity there is. And this will be important when we get to our plasma membrane, right? So saturated or straight, when you look at the, let's say this is a triglyceride, saturated fatty acid tails are straight, right? If we have a triglyceride and we have some unsaturated fatty acid tails, they're going to be bent. And this is going to make this a little bit more fluid because the tails can't get close to each other because they have a kink in them or they have a bend in them. Okay, so again, single bond unsaturated fats allow the fats to pack together tightly. And that's why they're a solid at room temperature. So when you think of saturated fats, you think of lard or butter, right? Animal products or animal fats. And that is, they're saturated fats, and those tails can get really close to each other because there's no double bonds, and so they're solid at room temperature. The double bond in unsaturated fats makes that fatty acid tail kink. And so you can tell if a fat is a unsaturated fat because it's a liquid at room temperature. So think of like vegetable oils like olive oil, grapeseed oil, what have you. Coconut oil is a little bit tricky. Coconut oil is actually a saturated fat, but it comes from a plant. But you'll notice that at room temperature, if it's cold enough, it's a solid, but if it's warm enough, it's going to turn into a liquid. So coconut oil is a saturated fat. It's just that its melting point is, I don't know, it's around, 80 degrees Fahrenheit or something like that. At the bottom, plants tend to be rich in unsaturated fats, and so these are going to sustain you longer, and they have more health benefits associated with them. So we want to try to eat more plants. Okay, here's a picture: butter saturated, solid at room temperature; oil unsaturated, liquid at room temperature. Let's talk about cis and trans unsaturated fats. Okay, what the heck is the difference? You guys have heard of trans fats, right? But let's get into why they're called that. So cis fats, or cis configuration, is when the hydrogen atoms are on the same side of the double bond. Excuse me. These guys occur in nature, right? So our bodies have evolutionarily designed to break them down. They're found in nature, and our bodies evolved to break them down. Trans fats are when the hydrogen atoms are on opposite sides of the double bond, and I'll show you a picture in a second. Trans fats are actually made, they're man-made, they're made up in a lab. They're not found in nature, so they're really hard for our bodies to break down. We store them in the liver, we'll store them to the artery walls, and they'll lead to plaque formation in the artery walls, and these guys are really dangerous. You don't want a lot of them in your diet. Okay, let's look at the picture. So cis fats, here's your carbon to carbon double bond. The hydrogens are on the same side. They're going in the same direction. So we call this a cis fatty acid. A trans fat is the H's are on opposite sides of the carbon-to-carbon double bond. And so these are not found in nature. If you heat like olive oil up really, really high, you can turn it into a trans fat. Margarine is a trans fat. The earth balance butters that you see, vegan butters, those are actually trans fats. You don't have to label it a trans fat if it has less than, I think, 0.5 grams per serving of trans fat. You don't have to label it per FDA regulations. So like all of your processed foods, like your Oreo cookies or your chips, they're all trans fats. They just don't have to label them. How do we make trans fats? We take vegetable oil, we add, if we pressurize it, and we add hydrogen gas until that causes hydrogenation, or causes the bond to break. You add hydrogens, and then it forces the bond to reform in an unnatural shape. When did we design these? Or what was the purpose of these? So when in World War II, we were trying to figure out how to get something that we could give to the troops that would have a really long shelf life. They took vegetable oil, did hydrogenation to it, and turned it into margarine. Take a look when you go to the grocery store. What's the shelf life of margarine? I think it's about 2 years. This is not good for you because these stick to the walls. These stick to your liver. These are really your artery walls in your liver. These are really hard for your body to break down. These are easy for your body to break down. Okay, here's a normal artery on the top, A, and B, is narrowing of the artery due to plaque formation. What is that plaque from? From trans fats, cholesterol, and high sugar in the diet. So the sugar is going to cause inflammation or locally acting chemicals to start this whole process. You're going to deposit the trans fats in this region, and cholesterol is going to stick to it. It kind of looks foamy. So if you could actually see this, if you look up plaque, it's really gross. It looks foamy and spongy. And then the endothelial lining, the innermost layer actually grows around it to sort of sequester it off. So it's kind of like a cyst, not really, but kind of. And you can see that artery is narrowed. And so it obstructs, it eventually can close, or you can throw a blood clot because of this artery being so narrow. Is it reversible? So this can lead to cardiovascular disease, obviously. It can lead to stroke. It can lead to blood clots in the coronary arteries, right? Is it reversible? It actually is. So let me tell you a couple of facts. Plaque streaks are found in children as young as 7 years old in the United States when you look at their arteries. In third world countries, because they don't have a lot of simple sugars and meats or fats, they eat a lot, or animal meats, they eat a lot of veggies and beans and lentils. They don't have as much plaque, if any, plaque formation. I will tell you also that they did CT scans of mummies from Egypt, and they found that they had plaque formations as well. So it's something in their diet that they're eating. They were probably eating meat because they were royalty. You can actually break down this plaque. So if you cut out your animal fats and your excess fat, excess cholesterol and sugar, you'll actually pull the stuff out of the artery and degrade it. So you can actually reverse cardiovascular disease to a certain extent. Okay, phospholipids are second category. Phospholipids, phosphate group in a lipid. So phosphate group 2 fatty acid tails. It's a diglyceride. Di means 2, right? Diglyceride 2 fatty acids, tails attached to a phosphate, glycerol. It has a hydrophilic head because it has a phosphate group, and then the fatty acid tails are hydrophobic. What does hydrophilic mean? It's water loving. What does hydrophobic mean? Water fearing. This is a main component of our cell membrane. Right, so here's a picture of a phospholipid. Here's your phospholipid, and here's your phosphate group. And notice it's a PO4, right? Non-lipid group containing nitrogen. And so this is the polar region. Here's your glycerol backbone, and then here's your fatty acid tails, right? These guys are non-polar, and these guys are polar. And so Um, these guys are used to make up your plasma membrane. So these are going to orientate themselves towards the water, and then these are going to orientate themselves towards the inside of the membrane. Here's a glycolipid. A glycolipid is actually a main component of our, well, you find it in our plasma membrane as well. These guys sort of embed themselves in the lipid area, and then this carbohydrate sticks out on the outside of the cell. So these are glycolipids. We have glycolipids and glycoproteins in our plasma membrane, and these serve as like cell recognition molecules, or protection against foreign invaders, or for our immune system to recognize our cells as our cells. Here's a phospholipid. So we can take phospholipids, stick them in water. The heads are polar, so they orientate themselves towards the water. The tails are non-polar, so they orientate themselves within one another. Here's the glycolipid, so they're going to stick out. So you can kind of get the idea that these guys are for cell recognition. Here's the phospholipid bilayer. So here's our plasma membrane. Here are the polar heads. Right? And then here are the nonpolar tails. And so the nonpolar tails sequester themselves among themselves because they're hydrophobic. They fear the water. They don't want to be around the water. The hydrophilic heads orientate themselves towards the water. And so, hold on a second, you guys. I'm going to pause this. And I'll be right back. Okay. So when we talk about phospholipids, they comprise our plasma membrane. So the polar heads containing phosphate, the phosphate group, orientate themselves towards a watery matrix on the outside and inside of the cell. And then the tails are hydrophobic or non-polar, and they make this membrane. So when I think of a phospholipid bilayer or plasma membrane, it is the heads, excuse me, these guys are like an Oreo cookie, like the cookies portions, and this is like the Oreo filling, the white filling. Okay. This is going to dictate what can go in and out of the membrane. So hydrophobic molecules, because there's so much of that Oreo filling, the hydrophobic molecules are going to be able to pass freely through there, because the bulk of the membrane is hydrophobic. Hydrophilic molecules are not going to be able to freely pass through. Okay, so glucose is hydrophilic. It is polar and large. It's not going to be able to get through that membrane just by going through simple diffusion. Ions like sodium aren't going to be able to just get through that plasma membrane. Eicosanoids are example of locally acting hormones, like prostaglandins, leukotrienes, and thromboxane. These guys are derived from arachidonic acid, which is an omega-6 fatty acid. They act as local messengers, autocrine and peregrine, for inflammation and immunity. They act on endothelial cells, platelets, uterine mast cells to trigger local responses. So they sort of act like local hormones, if you will. An example of this would be injured cells release prostaglandins to trigger nerve endings, so you feel pain. Here's what eicosanoids look like, or there's a picture of a prostaglandin, just so you can appreciate how complex it is. All right, the next category is steroids. Steroids are made up of 4 fused carbon-containing rings with variable side groups. What does variable side groups mean? It has a side group that can be many different side groups or compilation. Cholesterol is actually a precursor to all steroids. So you have to have cholesterol in your diet to make steroids. Cholesterol is actually used in your plasma membrane to help with stability. So those fatty acid tails, those double bonded fatty acid tails, make the plasma membrane more fluid. Cholesterol stabilizes it, and we'll talk about the plasma membrane a little bit later. These guys you have to obtain primarily from your diet. You can synthesize a little bit in there in your diet, in your body, but you have to obtain them from your diet. You use cholesterol to make up your steroid hormones. So example of those would be estrogens and testosterones. corticosteroids and calcitrol, vitamin D. Corticosteroids, when you think of those, think of cortisol, right? So these are stress hormones. Calcitrol is the active form of vitamin D, and it is going to help with calcium reabsorption, or excuse me, calcium absorption in the GI tract in your small intestine. And then bile salts. Bile is actually a fat emulsifier, so it's going to take a big fat droplet and break it up into little fat droplets. So if you think of laundry, no, like the Dawn commercials where they take some soap and drop it in a pan of oil, and the oil dissolves into little droplets. That's what bile does for our digestive tract. Here's a picture of cholesterol. So it's a 4 fused ring component with um, a hydrocarbon chain, and you can use it to make cholesterol, progesterone, androstenedione, which ultimately it's testosterone and estrogen. And so you use cholesterol to make up your steroids. Your steroid is a category, and that could be steroid hormones a lot of the time. Cholesterol is insoluble in water. So how do we carry it? We actually carry it using lipoproteins. Lipoproteins have 3 parts. You can read that. I'm going to show you a picture of it. So here is a lipoprotein, or an apolipoprotein, right? So here is a lipoprotein. Notice that is made up of phospholipids. So if you look at it closer, here's phospholipids, and then here's free cholesterol embedded. So little black dots are free cholesterol. And then you have these huge apolipoproteins. And then inside, in the core, you have triglycerides and cholesterol esters. We have very low density lipoproteins. We have low density lipoproteins and high density lipoproteins. You've heard of LDL and HDL, right? Let me explain what they really are. So low density lipoprotein is a lipoprotein that has a lot of fat and not much big proteins. Why is it called low density lipoprotein? Because it has so much fat, it weighs less, right? High density lipoproteins are called high density lipoproteins because they have a lot of protein and not much cholesterol and fat. So they're going to weigh more, right? What are the job of these guys? LDLs, or low density lipoproteins, take cholesterol and fats from your liver to your body, right? So let's say you eat a big fatty meal. Let's say you eat some French fries, right? The French fries are going to be broken down in your digestive tract, in your small intestine. You're going to pull them into the hepatic portal using very low density lipoproteins to the liver. The liver is going to modify them and stick them into low density lipoproteins and take them to the body cells. The body cells are going to pull them in and use what they need, and then they're going to get converted to high density lipoproteins and get kicked back to liver from the body tissues. You want more high density lipoproteins compared to low density lipoproteins because they are going to scavenge cholesterol from the body and return it to the liver. So you can't obviously tell how many you have in the blood supply. So what they do is they give you a ratio. So the LDL to HDL ratio is an index of the rate at which cholesterol leaves the cells and returns to the liver. A low ratio is good, meaning that there's less cholesterol, right, in the bloodstream. So when they do blood labs, and you should be having your blood drawn on a regular basis, so you know your normal values, your baseline values. This is a bad cholesterol, this is the good cholesterol. So you want a lower number because it's telling you that there's less bad cholesterol and more good cholesterol. I want you to know normal values, right? So total cholesterol, it's LDL versus HDL, or LDL divided by HDL. It's less than 200 milligrams per 100 mils of plasma or deciliter. HDL is the good cholesterol. You want it higher than 60 milligrams per 100 mils of plasma or deciliter of plasma. And then LDL should be less than 100 milligrams per 100 mils of plasma. It's 100 mils of plasma, or, oops, it's not going to let me do it. Or we can say this is 1 deciliter. Okay, just so you know. So if you hear me saying a deciliter, it's the same thing as 100 milliliters. So if you can picture that in your brain. Triglycerides, which should be the level of triglycerides, less than 150 milligrams per 100 mils of plasma. Okay, the next category are proteins. Proteins are the most abundant and important organic molecule in our body. If they're the most abundant, or let me back up. So they're the most abundant. They contain carbon, hydrogen, oxygen, and nitrogen. So these guys are unique in the fact that they contain nitrogen. When we use these guys for metabolism, we actually have to rip off the amine group or the nitrogen containing group because we can't just stick this into metabolism. So we ultimately, and I'm jumping a little bit into metabolism, but since I have you guys here, since you're kind of stuck, you have to listen anyway. This is an NH3 group, right? And you're going to convert that to ammonia. And that is toxic. And so your liver actually, your liver is what does this. You're going to convert it to urea. This is toxic as well, but it's not as toxic. Urea, I don't know where my eraser is. Let's see. It's probably going to erase the whole thing. Urea is less toxic, and so in that you can actually kick out in your urine from the kidneys. Okay. So I think it's actually NH2. Let's actually write this properly. So let me reset. It's NH2, so you don't learn the wrong thing. Plus. And then it becomes ammonia, which is NH3. That's toxic. You don't want excess ammonia because that can cross the blood-brain barrier and cause you to hallucinate, and then your brain will shut down. And so we convert it to urea, which is kicked out in the urine. Okay, so that's a little bit of metabolism, but let's get back to focusing on this. All right, so proteins, what do? They support, they provide movement, contractile proteins, right? They are carrier proteins, they transport stuff. They act as buffers, so they're going to keep the pH stable. They're going to regulate metabolism. So enzymes are an example of a protein that regulates metabolism. Coordination and control, hormones, and antibodies are defense proteins. I want you to think about our body is made up of mostly proteins. And so you want to think of that we would like to use proteins to break down for fuel because we have so much of them, but we don't. We use proteins last as a fuel source. Because our body is made up of proteins, we're going to not break those down, right? They have functions. I also want you to be aware of when someone is wound healing, they need more proteins because they need to repair those cells. And so if they're wound healing, you have to be careful. You want to make sure that a person is taking in more protein because otherwise they're going to pull it from the body. And in starvation, you know, if you're looking at adolescents or geriatric patients who have feeding tubes, you want to make sure that you're giving them enough proteins or that they're getting enough proteins, because otherwise they're not going to be able to produce enough protein hormones and they're not going to be able to make enough antibodies. And so a lot of times if somebody is going through starvation, whether it's intentional or not intentional, they die from secondary infections because they're not making enough antibodies. Or like, for example, my husband and I watched the Naked and Afraid shows, right? So there's two people, they're in a desolate area, and they're supposed to survive for, I think, like 20 days or something. And, you know, the first few days, they're fine. They can think, they're good. But then as they go day by day by day without getting in any food source, their body gets all of whack. And so they're releasing a lot of stress signals. They can't think properly. They start to break down. They start to hallucinate because they don't have enough hormones because they're not getting in enough proteins. They start to, you know, their body starts to break down its muscle. And so it's really dangerous. Proteins, the monomer or amino acids, there are 20 different amino acids that make up all the proteins in your body. Pretty crazy, huh? You can Some proteins in your body are like 200 different amino acids stuck together or like in a sequence. Some proteins in your body are like thousands. Okay. Most of those amino acids you can synthesize, but some of them you can't. And so guess what? They're called essential amino acids. They have to come from a diet. The complete protein has all of the essential amino acids in it. So for example, quinoa is a complete protein. Um, a lot of animal meat are complete proteins. Um, a lot of times indigenous cultures knew which food sources to stick together, which proteins to stick together to make a complete protein. So for example, when we think of beans, what do we think of eating with it? Rice, right? So that would be a complete protein, or, squash and corn and beans. That's a complete protein as well. So really interesting. A lot of the indigenous tribes, whether it's Native Americans or whether it's tribes in Asia or South America or Africa, already have this down. We just don't have all the science and analysis to back it up yet. So here's amino acids. I don't want you to know whether they're non-polar, negative, positively, and polar, and which ones they are. I just want you to be aware of them, right? So there are some that are negatively charged, some that are positively charged, some that are polar, some that are non-polar. The essential ones are highlighted in yellow. All amino acids have the same basic structure. So they have a central carbon atom, hydrogen atom, an amine group or amino group, NH2. This could be NH3. And again, we see the carboxylic acid group. That's a COOH or COO minus. The variable side chain or R group is different for each amino acid. So how do we get those 20 different amino acids? They all have a central carbon. They all have an H off of it. They all have a carboxylic acid group and an amino group or an amine group. Where are they different? So amino acid 1 versus amino acid 2 versus amino acid 3, et cetera, all have a different group or a CH whatever or R group or variable side chain right there. So let's look at the R groups just so you have an idea. So whatever that R group is, some of them are non-polar, some of them are polar, some of them are charged. These side groups are what gives the amino acid its chemical properties, right? So this is going to help the amino acid to fold. Let me show you a picture of this. Okay, so let's say I have a protein, and so here is, can I get this to work? Here's amino acid 1, here's amino acid 2, here's amino acid 3, and amino acid 4, right? Let's say this is no charge, this is positive, and this is negative, and this is no charge, okay? So that R group is positive, that R group is negative, this is nonpolar, this is nonpolar. When these guys start to fold, how are they gonna fold? The positive and negative are gonna be attracted to each other, and it's gonna start to fold like this, okay? If this was a negative group, right, and that was a negative group, what would happen? They would repel each other, so they'd sort of fold like that. So the charges on those R groups are going to help that protein start folding. Think about if it's an enzyme, right? It has a specific shape. If it's a hormone, it has to have a specific shape to bind to your cells to cause some type of change, and so that's dictated by those R groups. Excuse me. Here's examples of charged polar side chains. So you can see those R groups are very variable, right? Some of them are sort of small, long, ring structures, uncharged polar side chains like tyrosine, cysteine, serine, threonine. If you've taken biochem or biology majors courses, these guys should be familiar to you. Here's tryptophan. Where do we find tryptophan? Good primarily in Turkey, right? It supposedly makes you tired. So here's your COO. Here's your carboxylic acid group, your amino group, your H, and then right here would be this whole structure, tryptophan. Okay, just to give you an idea. I'm hooking these guys together requires a dehydration synthesis reaction that produces a peptide bond or a peptide excuse me. So the peptide bond is what forms. The peptide is the chain of amino acids. So let's say we want to take these monomers and stick them together. So here's glycine, here's alanine. We're going to use dehydration synthesis and an enzyme. It's going to force a bond between these two. And so this is a peptide bond. This is called a peptide. And so we can do a lot of, so let me back up. When you eat proteins, your digestive tract will break them down into these monomers, into these single units, right? And then you'll pull them into the bloodstream, you'll pull them into the cells, and then you'll build them up, whether there are enzymes inside the cells or receptors or some type of structural protein or contractile protein, you're going to build them up inside the cell. Let's talk about how they actually get made. So let's say I need more of a certain enzyme. I'm going to go to my DNA inside my nucleus. I'm going to decode the lagging strand to make a template or an instruction messenger RNA. And I'm going to go inside the cytosol and find a ribosome. And then the ribosome is going to plug and chug and make attach. So it's going to use a code from the DNA to come up with a sequence of amino acids. And so that's the primary structure. So the primary structure of a protein is a sequence of amino acids all along that polypeptide. The secondary structure results from hydrogen bonds interacting with one another of those amino acids. So they're going to form alpha helix or beta pleated sheets. So I'll show you guys these in a second. So tertiary structure, think of 3. It's going to be a unique shape. It's not functional yet in this step. And then quaternary structure would be a final protein shape, and this is going to be functional. This usually happens in the rough endoplasmic reticulum. This gets shipped to the Golgi, and then the Golgi is going to put it together to be functional and then ship it out. So it's either going to stay inside the cell or get kicked out of the cell. Here's a primary structure. So this is just an amino acid stuck to amino acid stuck to amino acid. Okay. These are called polypeptides because the covalent bonds are a peptide bond. Secondary protein structure, the polypeptide folds on itself in a regular pleated fashion or a regular fashion. The folding is going to produce helical structures like alpha helices. So think of like a ringlet or a curl in a woman's hair, right? It's a spiral. These are due to the amino acids hydrogen bonding with one another, or they fold in like accordion-like pleats. Or when you think of a paper fan, when you fold a paper fan, that's how it's a beta pleated sheet. These secondary structures are because of the hydrogen bonds between those amino groups from one amino acid to the next. Here's an example of an alpha helix, right? So here's amino acid 1, 2, 3, etc. And there's the hydrogen bonds. And if you remember back to chemistry, these hydrogen bonds are really weak. And again, they're dotted lines, so they're really weak, so they can break, but it's going to stabilize it a little bit. The tertiary structure comes from the interactions of the side groups. So if one amino acid is negative, one is positive, then they're going to attract each other. These interactions involve hydrogen bonds, but more so ionic and covalent bonds. So ionic and covalent bonds are stronger than hydrogen bonds. These side groups or those R groups interact with one another, and that's going to start causing that protein to fold. And so the tertiary structure is more folding than a secondary structure. It's still not a functional protein yet. So let's take a look at the tertiary structure. An example of this would be hemoglobin. Okay, so hemoglobin has 4 subunits. Here's the first subunit, the second one, the third one, and the fourth one. This is an alpha subunit, a beta, an alpha subunit, and a beta subunit. Okay, this is functional hemoglobin. This is a quaternary structure. So hemoglobin is an example of a quaternary structure. If you take out the subunits, right? So if you look at just one subunit is considered to be a tertiary structure. It's not functional yet. you have to stick them together to make a quaternary structure. But take a look. So here's a beta pleated sheet. Here's an alpha helix, beta pleated sheet, alpha helix. And then you see all these folds and whatnot. And that's due to the R groups. So the tertiary structure is folding into sort of a globular protein, but it's not quite functional. This occurs in the Golgi, and then this occurs in the Golgi. So sticking them together to make a functional quaternary structure. Quaternary structures, two or more tertiary structures stuck together, gives protein its final shape. Structure determines function. There are two different types of classes of proteins: fibrous proteins like keratin, or I'm trying to think. Collagen. So keratin, which is found in your hair and nails, collagen, which is underlying like in your connective tissue. These are fibrous proteins. Their structural sheets or strands are really strong, right? Globular proteins are more so what we talk about in physiology. These guys are spheres with active functions like hemoglobin. The function is based on the shape. The shape is dictated by the sequence of amino acids and how their R groups interact with one another. Here's a picture of a globular protein and a fibrous protein. I guess I just had to turn the page right. Okay, they do a lot of different important functions, you guys. Enzymes speed up chemical reactions. They regulate gene expression in cells. They're important structural components to cells, so they adhere cells to one another. And then they act as chemical messengers. An example would be hormones. Okay, so let's get into enzymes. How long would it take to digest this Carl's Jr. burger? Okay, or actually, how long would it take if you stuck it outside? No animals ran off with it. So bacteria and fungi ate it or broke it down. It would take probably, a month, a couple of months, right? How long does it take in your body to go from mouth to anus? Good, it takes about 8 hours, right? So theoretically, there's 24 hours in a day. You should be having 3 bowel movements. So 24 divided by 3 is 8 hours. So from mouth to anus, it should take 8 hours. So if you are not having 3 bowel movements, you need to look at your fiber. Your fiber intake and your water. We'll cover that in the nutrition assignment. So why does this happen faster? My point of this is why does this happen faster in our body? What do we have? Okay, so we have enzymes. Enzymes are going to catalyze reactions so that we can, do what we need to do, right? So enzymes are proteins that speed up the rate of chemical reactions, or they catalyze chemical reactions. They're going to break bonds, and they're going to liberate energy from those bonds, and we're going to harness that energy to either do work or make ATP. Enzymes lower the energy required to get a chemical reaction to go, okay? So again, we come back to the hydrolysis reaction. Here's another name: decomposition or catabolism in our body. This is what our digestive tract does. It rips apart macromolecules into small subunits, right? Hydrolysis, right? You're adding water, ripping apart the bonds. That would also be catabolism. A synthesis reaction or dehydration synthesis would be forming chemical bonds, so that would be anabolism. This is more so what your cell does. So your cell will pull monomers in and build up stuff or go through anabolism. Think of anabolic steroids, anabolism, building stuff. Dehydration synthesis or condensation is an example of that. So you are pulling water off, or you're forming water as a byproduct to synthesize that bond. How do the enzymes work? They decrease the activation energy of the reaction. What is activation energy? Energy required to get a chemical reaction to go. Enzymes catalyze metabolic reactions by decreasing this activation energy barrier, right? So take a look at this plot. Let me ask you a question about this graph first. This is our x-axis and this is our y-axis, right? Is this the independent variable or the dependent variable? Good, it's the independent variable. and it's the independent variable, which is the one that we're measuring or that we're changing. So progressive reaction would be time. So this is 0 minutes, 1 minute, 2 minutes, et cetera, et cetera. Y-axis is where we graph the dependent variable, and dependent variable would be energy in this case. Is this continuous data or discrete data? It's continuous data, so we're going to make an XY scatter plot. Anything versus time is always going to be continuous, so it's always going to be a line graph or an XY scatter plot. Okay, good. So let's look at this. So this is energy. Here's your reactants, and here's your products, right? In order to get reactants to products, this is how much energy we would have to put in. A lot of energy, right? Without the enzyme. Take a look though, with the enzyme, we don't have to put in, we have to put in like 1/4 of the energy. And so this is really cost effective for us. So reactants with enzymes can go to products with not much energy input, okay? And so we don't waste as much energy when we use enzymes. And technically some reactions wouldn't even go if we didn't have enzymes. So substrates are what the enzyme acts on. Enzymes decrease the activation energy by binding to the substrate and stressing the bonds so that you don't have to put as much energy in to break those bonds. The active site is the location on the enzyme where the substrate binds. So let's look at a picture. Here's an enzyme. Let's say this is a enzyme that breaks down sucrose, or excuse me. So here's sucrose. Here's your glucose and fructose. Sucrose is going to bind to sucrase. Sucrase is going to stress this bond and snap it, and then it releases them. What happens when a protein comes? So here's a protein, right? The protein is going to come and try to bind, and it can't because this enzyme is sucrase. It only breaks down sucrose. So these enzymes are specific for whatever they break down. This binding site is called the active site. And if you want this reaction to go faster, you can add more enzyme, or you can add more substrate, or you can heat it up to a certain extent. What if we want to inhibit this? We can block the binding site, so we can have something that sits on this active site so that, that sits on this active site so that the sucrose can't bind there. Or mercury blocks enzymes. Sometimes you need things to activate, or actually in your body all the time you need things to activate the enzymes. So those are called coenzymes, like ATP or vitamins or minerals make these enzymes functional. Okay, so this you can read. These guys show specificity in the reactions they can catalyze, right? So enzymes can be regulated. How can they be regulated? These guys need cofactors or coenzymes to become activated. In fact, all of our enzymes, probably 99% of them, need cofactors or coenzymes to become active. So let's take a look at these. What the heck are cofactors? Cofactors are ions of metal elements or organic molecules, like magnesium is a big one, ATP is another one. Coenzymes are cofactors that are derived from vitamins, okay? So what do we use these guys for? These cofactors and coenzymes have to bind to enzymes to make them work, okay? So these guys have to bind to enzymes to make them work. Where do we get these guys from? We get these from primarily vegetables and fruits, right? So vitamins and ions, right? So when you think of ions, you think of electrolytes, you think of like calcium, magnesium, potassium. The ions, yeah, you can go out and eat dirt, but they're found in root vegetable, right? So when you think of coenzymes and cofactors, I want you to think of fruits and veggies. So what happens if you don't have enough fruits and veggies in your diet? Are you able to get your enzymes functional? No, you're not. So think about 0 to 5 when kids are developing most, and think about your geriatric patients, right? What if, so for example, in San Bernardino, we live in a food desert. Most people get their fruits and vegetables from 7-Eleven, right? You can get them at the dollar store too. Most people get them at 7-Eleven. And so if we're not getting enough fruits and vegetables, are enzymes functional? No. And so from 0 to 5 is when you're laying down bone and body, right? So if a child isn't getting enough fruits and vegetables to activate their enzymes, are their enzymes functional? No, they're not. And so are they developing properly? No, they're malnutritioned and they're not developing properly. And so think about long-term consequences of malnutrition in 0 through 5. If they're not getting enough nutrients, is their brain developing properly? Is their body developing properly? Are they going to have health issues in the future? Are they going to have disabilities? Cognitively, are they going to uh, be functional members of society? Are they, we want to think about taking care of our community so that we, have functional members of society. So it's really important through 0 through 5 in geriatric people to have enough fruits and veggies. How many fruits and veggies should you have per day? 7 to 9 servings. So a cup is a serving of veggies, half a cup is a serving of fruit. You guys are going to do a nutrition assignment in a day or so, so that's going to help solidify this. Really important. I can't stress how important nutrition is, guys. Enzymes can only work as fast as they can. You can actually saturate them, right? So enzymes have a maximum velocity. So take a look. This is substrate concentration. This is velocity. So how fast this enzyme is working. You add some substrate, it has a certain plug and chug speed, right? You add more substrate, it plugs and chugs faster. You add more substrate, it plugs and chugs faster, it goes faster. Eventually, it's going to max out. So you add so much substrate, it's going as fast as it can to break down that substrate, but it can't go any faster. You've hit the maximum velocity of that enzyme, or we call that a saturation of an enzyme. These guys can be inhibited by heat. So heat is going to cause these to unravel. Changing the pH is going to cause these guys to unravel too, right? So think about a burger. You heat it up, it's going to cause it to change form because you're heating it up. When you stick fish or when you marinate chicken or what have you in lemon juice, right, or beer, or vinegar, it's acidic. That pH is going to actually unravel the protein as well. So it's going to chemically cook it. This is really dangerous for our bodies. So when you think of our bodies, when we have a high grade temperature, why is it so dangerous? You're heating up the proteins in your brain, right? You're starting to denature them. Why do we regulate blood pH so heavily? Because you need blood pH to be a certain level. You need your body pH to be a certain range, a narrow range, so that you don't unravel your enzymes, so that they stay functional. You can activate enzymes by, coenzymes, cofactors, usually magnesium, ATP, your vitamins, right? Like your niacin. You'll see a lot of, like, if you look at the processed food, they have a lot of vitamins and minerals added in for that reason. It's better to get it in a whole food than having it added in, like in cereal or, white bread. But you can actually inhibit these guys by binding an inhibitor. So molecules, sometimes pathways are turned off by excess of one end product of metabolism. Heavy metals, so mercury is so dangerous because it binds to enzymes and makes them not functional. Different types of drugs or inhibiting drugs are gonna block these guys. Enzymes, when I talk about carrier proteins are globular proteins, and because they're globular proteins and enzymes are globular proteins, they all behave the same way. Coming up to our last category, nucleotides are large organic molecules that form genetic material of the cell and are used in energy transfer of the cell. I want you to read this portion in your textbook so that you have a clear understanding of nucleotides, because it can be confusing. Nucleotides comprise DNA, so deoxyribonucleic acid, DNA, is made up of nucleotides. Ribonucleic acid, RNA, is made up of nucleotides. Adenosine monophosphate, which are an ATP, adenosine triphosphate, and cyclic adenosine monophosphate, cyclic AMP. These guys are all energy transfer molecules. So they're sort of variations on a theme of a nucleotide. Nicotinamide adenine dinucleotide, which is NAD+, and flavin adenine dinucleotide, which is FAD+. We're going to talk about those when we get to metabolism. Those are cofactors for enzymes. The generic general nucleotide structure, kind of like the generic amino acid structure, right? You're always going to have a nitrogenous base bound to a sugar and bound to a phosphate group. These nitrogenous bases can vary. So they can be adenine, thymine, cytosine, guanine, or uracil. The sugar can be a ribose sugar or a deoxyribose sugar, and there's always a phosphate. This, when we're talking about DNA, think of DNA as a ladder. These are going to be the sides of the ladder, and then the nitrogenous bases bind to one another to form the rungs. Adenine is one choice for your nitrogenous base. It's classified as a purine. Guanine is classified as a purine as well. Purines are double bonds. I'm not going to ask you the structure, but I want you to know that adenine and guanine are two nitrogenous bases. Two options, I guess. Pyrimidines, cytosine is a pyrimidine, thymine and uracil are pyrimidines. So these can be your nitrogenous bases. Uracil, so if you think of DNA, deoxyribonucleic acid is found in the nucleus. It stays in the nucleus. Uracil is the RNA component, right? So uracil replaces thymine when you're making RNA. So uracil is found in RNA. Thymine is found in DNA only. Sugar options. 2-deoxyribose. It's the first, second carbon. So 2 denotes what carbon. And this carbon has a deoxy or no oxygen. So 2-deoxyribose or deoxyribose is a sugar used in deoxyribonucleic acid, DNA. So this is a sugar found in DNA. The ribose is a sugar used in ribonucleic acid. Ribonucleic acid, this is a OH at the second carbon, and this is a ribose. So let me back up. RNA uses a ribose sugar, and DNA uses a deoxyribose sugar, hence the name. That's how we got the names. Deoxyribonucleic acid, DNA, is 2 strands, a complementary chains of nucleotides. A nucleotide, again, is a phosphate bound to a deoxyribose bound to a nitrogenous base. Your choices for nitrogenous bases are what? Adenine, thymine, cytosine, or guanine. Okay, not uracil because we're not talking about RNA, we're talking about DNA. So how do nucleotides work? You have a phosphate group, PO4, bound to a sugar, which is a deoxyribose because we're talking about DNA. And then that deoxyribose can be bound to adenine or guanine or thymine or cytosine. Hi guys. Shh, 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 shh. And so the trick or the rule is that adenine always binds with thymine. So if we're talking about DNA, DNA has one strand that's going in this direction, and then another strand that's going in this direction. This is your sugar phosphate backbone and sugar phosphate backbone. And then you have your A off of here, and the complement to A is what? Adenine, thymine. So thymine is here. How do we know it's adenine and thymine together? They double bond. They have 2, hydrogen bonds. Okay, then let's say this next nucleotide was a G or guanine. What is a complement to that? Guanine always binds with cytosine, so that would be cytosine. And they have 3 hydrogen bonds together. Okay. This is how our DNA works inside the cell. So we call this complementary pairing. Let's say we wanted to use this so that we could make an enzyme. So we're going to go inside the DNA. We're going to zip these guys apart, and then we're going to make a copy of this guy. Let me see if I can get, pen color. So it would be, let's do green. It'll be like Christmas colors. So if we're talking about making a copy of this for messenger RNA, it would be what binds with T? Good, adenine. What binds with C? Guanine, okay? So we would then make messenger RNA complement to this, it's called the lagging strand, and then this would go out in the cytosol or into the rough endoplasmic reticulum, find a ribosome, and decode it into ultimately a protein, okay? I'm not going to ask you all the details. You actually will cover this in microbiology, or if you take, I think you cover it in biochem, and you cover it in majors biology as well. Okay, I just want you to know that A always bonds with T and C always binds with G. How do we know that this is A and T? Because there's 2 hydrogen bonds. How do we know that this is C and G? Because there's 3 hydrogen bonds, okay? This is what DNA looks like. DNA stores genetic information in the form of genes. Your DNA makes up your genes make up your chromosomes. We have 23 pairs of chromosomes in each cell. What is the function of DNA? To duplicate for cell division so you can make more cells, or to use as a template for protein synthesis. DNA carries our inherited characteristics and controls enzyme production and metabolism. Ribonucleic acid, RNA is going to take the code from DNA and translate it into proteins. It's single-stranded, has a ribose sugar instead of deoxyribose, and contains uracil instead of thymine. So if we had a G for DNA and we were making RNA, we wouldn't put a T there, we would put a U there. mRNA is going to take the code from DNA, translate it into proteins. So we call this transcription and translation. So think about inside the nucleus, you have the DNA, you are transcribing the DNA code, because you're not going to take the DNA out into the cell and make proteins from it. So you're going to take a copy of it, right? Instruction template, and then you're going to go and translate it from one language into another. So you're taking RNA and translating it into proteins. So messenger RNA, transfer RNA, and ribosomal RNA are the 3 different types of RNA. On the right-hand side is DNA, left-hand side is RNA, and you'll notice that the colors match up. So it's color-coded. So you can take a look at that. Notice instead of, let's see, where's our T? So notice instead of thymine, here's your leading strand, right? And here's thymine. This would be guanine, right? Here's our lagging strand. So if we're going to make a complement to this with RNA, we're not going to use thymine. We're going to actually use uracil instead of thymine, or in place of thymine. Nucleosides are variations on that theme. They're used to store energy, transfer energy, or as in a messenger. Take a look at the bottom one. Adenosine triphosphate is ATP. It has 3 phosphate groups. Let's take a look at the picture. So here's your adenine, which is the same as your nitrogenous base, right? And then you have your ribose sugar, and then you have a phosphate group attached to it. Notice this phosphate group is negative. And then for ATP, which is a high energy molecule, we call ATP adenosine triphosphate. How many phosphates does it have? 3. 1, 2, 3. So that's your triphosphate. Why is it so high energy? Let's take a look. Here's a phosphate group, and then you stick another phosphate group next to it. They're negative. What happens when you stick a negative next to a negative? They want to repel each other. And so in order to get this bond to form, you have to use some energy, but it traps a lot of energy in this bond. And then not only that, you have the other phosphate group, which is a -2, and you shove that against a negative and a negative. So you shove that right next to it. So it's a really high energy bond. So ATP is so high energy in the body, and it's your energy storage molecule because of these phosphate groups stuck together. And then when you want to do work inside the cell, ATP is going to bind to your enzymes, and then your enzymes or carrier proteins or whatever are going to split off this phosphate group, and you're going to release a lot of energy, right? And then your carrier protein or whatever you're trying to do is going to grab that energy. So you toggle between ATP and ADP. So ADP is lower energy, ATP is higher energy form. Let's see. So an ATPase is an enzyme that catalyzes phosphorylation, excuse me. So an ATPase is an enzyme that makes ATP. Usually ATP synthase makes ATP. ATPase uses ATP. FAD and NAD are electron carriers. Cyclic AMP is sort of a secondary messenger system component. Take a look at NAD. So NAD is an electron carrier. Really cool. Look how complicated it is. It's really an interesting molecule. And then here's cyclic AMP. We'll talk about NAD more when we get into metabolism. Okay, yay, we're almost done. Micronutrients. Micronutrients are substances that we need in small amounts by the body. So vitamins and minerals. Is it best to get it in pill form or in fruit and vegetable form? Fruit and vegetable form, obviously, because you get more nutrients per fruit or vegetable, right? So a banana doesn't just have one nutrient, it's made up of several. The rule is, if you have no other options, take a pill. In the pill form is better than nothing. But you want to focus on when you're eating to have a variety of colors, because a variety of colors means a variety of micronutrients. These guys have antioxidants in their skin, so all these different colors mean different nutrients that you're getting in those skins, and different nutrients that you're getting inside the fruit or vegetable. So you always want to sort of try to aim for a rainbow of colors when you're eating. And it's easier said than done. I don't always do that either. Okay, vitamins. There are 13 essential vitamins. What does that mean? You have to get them from an outside source. Most can't be synthesized from the body. D and K are the exceptions. Do I want you to know that? Yes. So D and K you can synthesize by the body. Everything else you have to get from an outside source. These guys are functioning as coenzymes, so they activate enzymes. And you can imagine that since they activate enzymes, when you don't have a vitamin, you're not going to be able to carry out chemical reactions. Very dangerous. I want you to know how vitamin D is made. So it's synthesized by sunlight. What do we use vitamin D for? It pulls calcium in from the small intestine into the bloodstream. So we need vitamin D for calcium absorption. You can get it via sunlight. You can get it, it's usually supplemented. It's put in dairy products. Here's an animation. So sunlight hits your skin, converts it to vitamin D3. D3 is going to go to the liver. It's going to convert it to a different form of vitamin D3. It goes to the kidney. The kidney is going to convert it to a different form of vitamin D3. This is the active form. It's going to pull in calcium from the digestive tract. You can get it from supplements and fish and meat. Most people in the United States are vitamin D deficient, usually elderly and or geriatrics and babies because they don't get enough sunlight. How do you get enough vitamin D? Sit in the sun, 20 minutes 3 times a week usually does it. Vitamin K is required for blood clotting. Vitamin K is actually not produced by our cells, but it comes from bacteria living in our large intestine. These bacteria love green leafies. Green leafy vegetables have these guys go crazy and break down those green leafies. And you do have vitamin K in some of those green leafies. So the vitamin K is going to go to the liver. The liver actually uses that vitamin K to make clotting factors. So clotting factors to form blood clots. So if you have an issue with making clotting factors, sometimes it's because you can't absorb vitamin K from the digestive tract. If you make too many clots, you want to decrease clotting by using an anti-clotting drug like Coumadin or warfarin. Patients who are on Coumadin or warfarin, who are on anticoagulants, should not eat a bunch of green leafies because the green leafies are going to inhibit, they're going to inhibit the drug. Okay, I'm going to pause for a second because I'm running out of battery. So I need to go plug it in. I'll be right back, guys. Let's see. Okay, so I'm back. So let's see. So, okay, so hydrophilic vitamins, because they're hydrophilic, oops. Hydrophilic vitamins are water soluble, so they can be dissolved in the plasma. They're not stored in the body. They are excreted in urine. So think of vitamin C, right? Hydrophobic vitamins are not soluble in water, so they can be stored in our body fat. So it's possible to have excess of these, resulting in toxicity. So I want you to know these guys, A, D, E, and K. Take a look. So you don't have to know these tables, but I want you to appreciate deficiencies, right? So here's your B vitamins, panthenoic acid, biotin, C. Take a look. Functions, required component of many enzymes. Iron absorption, required component of many enzymes. What happens if we don't have enough of vitamin Bs, right? And vitamin C? Water retention, heart failure, skin lesions, neural tube defects, right? So you know that if a woman is pregnant, she needs to take excess folic acid. Why does she need to take excess folic acid? To make sure that her spinal cord actually closes. So the tube closes around the spinal cord so that the spinal cord doesn't develop outside of the body. And if that does happen, that's called spina bifida. Anemia occurs. A lot of your B vitamins, anemia and nerve disorders, because your B vitamins are required for proper nervous function. Vitamin C, scurvy, and poor wound healing. So you can take a look at those guys. Where do we get them from? You can get them from leafy greens. You can get them from legumes, whole grains, veggies, nuts. Those are good for vitamin Bs. Fat-soluble vitamins, A, D, E, and K. Take a look at where we get them from. Here's a function. So A is eye pigment and immunity. So A is for immunity, D is for calcium absorption, E is a component, a coenzyme, and K is for blood clotting. If you don't have enough of them, here you go. Skin sores, blindness, bone deformities, because you're not getting in enough calcium, birth defects, anemia, bruising, abnormal clotting, and severe bleeding, effects of excess. So because they're fat soluble, we can have excess of these guys. So kidney damage, diarrhea, vomiting, fatigue, weakness, headache, diarrhea, liver damage, and anemia, and drowsiness, and bone pain and hair loss. Minerals. These guys they're inorganic. They don't contain carbon, but we have to have them for normal cell functions. When I think of minerals, I think again of ions or electrolytes. So we need them for proper fluid balance, muscle and nerve function, and building bones and teeth. We'll talk about these guys when you get into excitable cells. Examples: calcium, chloride, sulfur, magnesium, phosphorus, and potassium. Magnesium is a big one to activate enzymes. Here's minerals. So there's your minerals, guys. Where do we get them from? Functions: bone strength, blood clotting, acid in stomach, required component of enzymes, bone and tooth formation, water balance, muscle function, water balance, nerve functions, components of many proteins. Okay, what if we don't have enough of them? What if we have excess of them? Okay, we'll talk about potassium. So when we get into excitable cells, you guys will learn that potassium, not enough potassium in the plasma, is going to cause fatigue and muscle weakness. Excess potassium is going to cause, it can cause hyperexcitability, so paralysis and excitable heart or heart failure. Antioxidants bind free radicals. So when you do chemical reactions in your body, sometimes those electrons get knocked out of orbit and they become free radicals. So they're bouncing around unattached to anything. Antioxidants actually bind the free radicals and prevent them from doing damage. Vitamins and minerals are antioxidants. You guys know these, beta carotene and carrots, lutein in spinach and kale, right? Lycopene in tomatoes. Again, this comes in from a lot of times from the skin of your fruits and veggies, right? So you want to eat a rainbow of colors so that you get enough antioxidants. Selenium, you need small amounts of it, but you find it in nuts. Vitamin A, vitamin C, vitamin E. I'm trying to think. Oh, sweet potatoes, right? My munchkin loves sweet potatoes right now. Okay, we're at the end of this. I will see you next time.